Hey everyone, welcome back to Window Seat Studies. We have been learning about histone post-translational modifications, in which we learned about histone acetylation in detail, as well as we learned about histone methylation. So, this is the continuation of histone methylation part, and today we will be learning about certain methylation marks. And today's topic of focus will be the H3K9 methylation mark. So we have learned that the histone modifications they they can directly perturb the chromatin structure or they can recruit certain factors which will be changing the chromatin environment. So we have learned all of this till now, and in histone methylation we learned that the methyl group can be added on the lysine residue or it can be added on the arginine residue. So on the lysine residue, it is added by the enzymes which are known as HKMTs, histone lysine methyl transferases, while the arginine methylation is done by PRMTs, protein arginine methyl transferases. So the HKMTs they can either monomethylate lysine residue. It can they can do dimethylation or trimethylation of lysine residue, while the arginine residue can be monomethylated or dimethylated. And the addition of the methyl groups, the two methyl groups, they can be added in either symmetric fashion or in asymmetric fashion. So this is what we have learned till now in the histone methylation part. So today we will be learning about the histone methylation mark, and that is H3K9 methylation. Okay, so this is the topic of focus for today. So the methyl group is added on the ninth lysine residue of histone 3 protein, H3 protein. So this is written in this way, H3K9 methylation. Okay, and this is done by the enzyme which is known as Suv39h1, suppressor of variegation. This was the first HKMT which was identified in 2000. Okay, so this was the first histone methyl transferase which was identified by the researchers. So SUV39H1, this is responsible for methylation of the ninth residue of the H3 protein. So this mark is repressive mark, means it leads to the gene silencing. So characteristic, it has a very special characteristic, and that is it is considered as hallmark of heterochromatinization. Okay, so this leads to the heterochromatinization. So we will be learning about detail why this is considered as a hallmark for the heterochromatin. So, we have, unlike histone acetylation, which directly perturbs the chromatin environment, the methylation, specifically H3K9 methylation, this this acts as a signal for the recruitment of certain factors. So this doesn't directly change the chromatin environment. So for example, in the histone acetylation, we learned that the addition of acetyl group, this neutralizes the positive charge of histone. So the neutralization of positive charge of histones weakens the DNA and histone interaction, right? So addition of acetyl group is directly responsible for changing the chromatin environment. Okay, so due to the addition of acetyl group, histone DNA interaction, they are getting weak, and this weak interaction is leading to the more exposed DNA. So it is making DNA accessible, and if the DNA is accessible, then we will assume that we will consider this acetylation as an active mark because it is leading to the activation of the gene. Okay, it is making the DNA more accessible. But unlike acetylation, histone methylation, and specifically H3K9 methylation, it doesn't directly change the chromatin environment. It doesn't directly changes the conformation of histone. It actually acts as a recruitment signal. So it acts as a recruitment signal, and it so this due to that it provides a docking site for certain factors Okay, and those factors will change the chromatin environment. So we will learn about those factors as well. So this H3K9 methylation acts as a recruitment signal. So it acts as a docking site for the protein which is known as HP1. This is heterochromatin protein 1. Okay, and this heterochromatin protein 1 is able to bind to the H3 if it is methylated as the ninth residue. So whenever there is H3 and it gets methylated, so okay, so it gets methylated, so on the ninth residue, and this is done by Suv39h1. This is done by Suv39h1, suppressor of variegation enzyme. So it adds the methyl group on the ninth residue, and is the this provides docking site for HP1. So this HP1, this protein has a domain which is known as chromodomain. Okay, and HP1 is a protein which has a chromodomain, and this chromodomain this binds to the H3K9 methylated mark. So uh, so here's a thing that you have to remember always, that the proteins which has chromodomain, they are able to bind to the methylated histones. Okay, so they are able to bind to the methylated histones, while the proteins which has bromodomain, they are able to bind to the acetylated histones. Okay, so this is a very important point that you have to always remember. So here you can see that histone acetylation. Okay, so histone acetylation can directly perturb the chromatin environment because it is changing the charge on the histone protein, as well as it provides a docking site for the proteins which has bromodomain. So it works in both fashion. The mechanism of working of histone acetylation is that it can either directly bring the conformational change in histone, so it can directly alter the DNA histone interaction, or it can recruit certain factors which will be changing the chromatin environment, so which will be either activating or repressing it. In the case of histone acetylation, it is always the activation. But in the methylation, it doesn't directly change the chromatin. It provides a docking site for the proteins which has chromodomain. So this is a very important point, that chromodomain have the proteins which have chromodomain, they bind to the methylated histones, while the bromodomain containing proteins bind to the histone acetylated histones. Okay, so this is very important point you have to always remember. So this is the image which I have taken from a research paper which was published in Nature. If you want to read it in details, I'll put the DOI in the comment section or the description box below. So here you can see this is the chromatin. These are the nucleosomes. So around which the DNA is wrapped. So assume that this is the in this octamer that this is the histone 3. So on the histone 3, the methylation mark is being added by Suv39h1 methylase. So this is an HKMT, okay, histone lysine methyl transferase, which transfers the methyl group on the ninth residue, okay, on the ninth lysine residue. So it does the H3K9 methylation. Okay, so this enzyme is adding the methylation mark on the histone, and due to the addition of methylation mark, it provides a docking site for the protein HP1 protein, heterochromatin protein 1. So this protein, this is HP1 is responsible for heterochromatinization. Okay, so HP1 is responsible for heterochromatinization. So SUV39H1, it goes on adding methyl marks on the consequent histones. So first it adds on the first histone, then other histone, then another histone. So it goes on adding the methylation marks, and due to the addition of methyl marks, the HP1 comes and it recruits to those histone, and due to the HP1 binding, it the that that HP1 coated chromatin gets heterochromatinized. So this schematic shows the spreading of silenced and HP1 coated heterochromatin. So this is how it silences the gene. This is how it condenses the chromatin, and chromatin condensation leads to the silencing of gene. So that is why we consider H3K9 methylation mark as a repressive mark. Okay, so this is considered as a repressive mark. But this heterochromatinization, this is not indefinite. Okay, so this doesn't continue. So this has a limit, and that limit is decided by the boundary elements. Okay, so the boundary elements, they set the boundary between the heterochromatin and euchromatin. So here you can see beyond the boundary element there is active chromatin, so which is acetylated. Okay, acetylation is active mark. We have already learned about it. So this is how the boundary. So till the boundary element, the SUV39H1 methylase can work, and so that is why the boundary element, it defines the boundary between the heterochromatin as well as the active chromatin. So this is how H3K9 methylation is the repressive mark, and this is all about H3K9 methylation for today. In the next lecture we will learn about the very interesting phenomena of self-propagating loop. So this is it for today. Then we will also learn about other methylation marks such as H3K20 methylation, H3K27 methylation. Till then, see you. Bye bye.